Hi, everyone. So let's just dive right into this. Today, we're going to talk about something you absolutely have sitting in your medicine cabinet right now, but I bet you've never really thought about how it got there. We are unpacking the validation of semisolids, which is basically just the hardcore science of ointments and creams. Seriously, manufacturing pharmaceutical creams is exactly like following this flawlessly executed, extremely high-stakes recipe where millions of dollars and, more importantly, patient health are on the line. I'm going to break this entire process down for you so it's super easy and quick to learn. Let's get into it. Ever actually wonder how your medical creams are mixed so perfectly every single time? Think about it for a second. When you squeeze a little tube of medicated ointment, the active ingredient inside has to be distributed with total precision. Literally, the very first drop out of the tube needs to be exactly as potent as the absolute last drop. So it seems like a basic, simple tube of everyday cream actually requires some intense scientific precision, rigorous testing, and a super strict regulatory process that we call validation. Let's break down how they actually pull this off. First off, what exactly are we working with here? Well, in the pharma world, we call these things semisolids. That covers dosage forms like ointments, creams, pastes, gels, and rigid foams. Now, the official documentation uses this incredibly technical term, plastic rheologic behavior. But honestly, don't let that intimidate you. All that fancy jargon means is that the cream has the ability to cling to a surface and hold its shape as a little dollop until an outside force acts on it. So in really simple terms, it stays in a nice little peak on your finger until you actively rub it into your skin. Pretty intuitive, right? Okay, but before we can even think about mixing our cream, we have to prep. You can think of this pre-validation phase as the ultimate kitchen prep. Imagine you're a master baker about to compete in a world-class pastry competition. You wouldn't just throw your flour into a dirty bowl, right? Obviously not. In pharma, companies have to ensure their kitchen, their ingredients, and their tools are flawlessly prepped. We're talking strict cleaning validation, verifying every single raw material, making sure the operators are fully trained, and calibrating every piece of equipment. The whole shebang must be locked in, qualified, and documented before a single drop of product is ever made. So, our kitchen is spotless, our tools are calibrated, and the ingredients are totally ready to go. The big question now is, how do we actually mix the flawless batch? This is where the actual cooking process begins, and let me tell you, it is a beautifully orchestrated sequence. Here is how it goes step by step. First, we prepare what are called the internal and external phases to create a completely clear solution. Second, we mix those two phases together. The big goal here is homogeneity, basically just making sure it's uniformly blended. Third is the homogenizing stage, where the heavy machinery really kicks into gear to establish the final appearance and that nice, smooth texture. And finally, the fourth step is the final mixing. This is where the scientists dial in the exact pH, the viscosity or thickness, and the assay content, which is a fancy way of saying they make sure the actual medicine levels are absolutely spot on. It's all about step-by-step precision. Now, there are kind of two sides of the exact same coin when making these creams. On one hand, you have the critical processing parameters. These are the chemical and physical properties we just talked about, getting that clear solution, hitting the perfect texture, the right pH, and the right viscosity. But to actually hit those chemical goals, you absolutely have to master the mechanical settings, which are the equipment critical process parameters. You got to precisely balance things like the mixing speed, the heating and cooling times, the flow rate, and even the vacuum pressure inside the giant vat. It's exactly like figuring out that you need to balance your baking ingredients with the exact oven temperature and mixing speed to get a flawless cake. And here is a massive, incredibly rigid regulatory rule that you absolutely need to know: 10%. When a company is doing an ongoing validation study to prove to regulators that their manufacturing process works, their batch size can only change by a maximum of 10%, and they have to use the exact same equipment. You can't just suddenly decide to double the recipe or use a completely different blender for fun. The FDA and other regulators demand absolute, total consistency, no exceptions. Let's expand a little bit on those strict rules for a validation batch. A validation batch is basically the ultimate test run to prove to regulators that your process works flawlessly before you're allowed to sell it to the public. Prospective validation is required for any new products or product transfers. And during these trials, the manufacturing process, the formula, and the equipment are completely fixed. No improvising allowed at all. Also, your validation batch size literally has to equal the commercial production size. And while you do need to use different lots of active ingredients, just to prove your process can handle slight natural variations, they still absolutely must come from the same approved manufacturer. It is super important to understand the difference between this validation testing and everyday routine testing. Validation testing is intense. It involved deep, bulk, and finished good testing based on strictly set release criteria. You are proving the entire process is fundamentally sound from the absolute ground up. On the flip side, routine QC release testing is what happens every day after a product is already approved. Routine QC involves testing standard samples separately just to make sure today's batch is as good as the master validation batch. The easiest way to think about it is this. Validation proves you can make it right. Routine QC proves you did make it right today. Okay, so once the cream is fully blended, we have to actually get it into the little tubes. And yes, the packaging phase is validated just as intensely. For container filling, the rules require taking at least 2 portions from different bulk trials and filling them into 2 separate batches. And this next requirement really shows how incredibly thorough regulators are. You actually must dedicate 1 entire bulk batch strictly to filling your smallest container, just to prove your machinery can handle its maximum possible continuous runtime. You also have to include studies on both the smallest and largest containers of the same type, which guarantees the fill weight is perfectly accurate no matter the size of the tube. Actually, let's back up just one quick second. We've mixed this massive multi-ton vat of medicated cream, and it looks great. But how do we actually sample it to prove to the authorities that it's uniformly exact all the way through? I mean, you can't just scoop a handful off the top and call it a day, right? The sampling process is honestly a highly strategic hunt. First, technicians have to take at least 10 samples that accurately represent the top, the middle, and the very bottom of the mixing vessel. Second, they specifically target the areas right next to the mixing blades, the baffles, and the shafts. And third, they actively seek out the bottom of the tank and known dead spots. Regulators are literally hunting for unmixed or undissolved material in the absolute hardest-to-reach corners of that giant vat. The logic is totally sound, though. If the tricky dead spots are perfectly uniform, then the whole batch is flawlessly blended. Now let's talk about the number 24. This is a very specific default rule in pharmaceutical manufacturing. If a company doesn't have sufficient supporting data, 24 hours is the default maximum hold time. A bulk batch can just sit there before it must be filled into its final packaging. So what exactly is a hold time? Well, imagine you mix this giant vat of cream on a Friday afternoon, but the packaging machines unexpectedly break down and aren't fixed until Monday. Will that cream separate or degrade or grow bacteria while it's just sitting there waiting? Hold time qualification is the science of figuring that exact thing out. It means running a test where you simulate actual real-world conditions by holding 1 full-scale bulk batch in the vat for the maximum practical time before you fill it into the tubes. You are scientifically proving exactly how long your finished cream can safely sit waiting without losing a single drop of its quality. We are in the final stretch now. During the actual mixing, operators are constantly doing in-process monitoring. They're recording temperatures, checking pump speeds, and keeping an absolute eagle eye out for any weird lumps, foam, or discoloration. Then, once the product is finally pumped into the tube, it enters finished product testing. This is where the really intense chemical and microbial lab tests happen. They check the net contents to make sure your tube isn't secretly half empty, they test for content uniformity, and they meticulously verify the exact pH and viscosity. Literally everything has to pass flawlessly. And that brings us to the definitive bottom line, the ultimate unbreakable rule of pharmaceutical manufacturing. You could have the most beautifully textured, precisely formulated, completely sterile cream in the entire world. But the product may not be released to the market until the validation report is strictly approved and officially issued, period. No matter how good the physical product looks in the tube, it stays locked up in quarantine until every single page of the paperwork, the testing data, and the cleaning validation is officially signed off by quality assurance. Paperwork is king. Okay, let's wrap this all up. The next time you reach into your medicine cabinet and use a simple everyday medical ointment, I really hope you take a second to appreciate it. Will you look at it just a little differently now? Because what seems like a totally basic household item is actually the result of extreme high-tech science, flawless engineering, and insanely rigorous validation. It is a massive, massive journey from raw ingredients to that exact little dollop on your skin. Thank you so much for joining me on this quick, easy learning journey to uncover the science of semisolids. Keep asking questions, keep learning, and I'll catch you on the next explainer.